Are you able to see my presentation?
Yes.
Okay, so good afternoon. In the last lecture, we studied out the in vivo spectroscopy, and we essentially covered hydrogen spectroscopy. We looked at the spectral parameters, and then saw the application of hydrogen spectroscopy in characterization of lesions, the brain lesions or lesions elsewhere in the body, and then also how we can categorize or characterize the tumors, whether the tumor is grade 1, grade 2, or grade 3. And then we also looked at how spectroscopy can be useful in early detection of breast cancer and the prostate cancer. And we showed last time in for the prostate cancer, in a healthy prostate, there's a very huge peak from citrate, and when the prostate is cancerous, the citrate peak is missing, and then there's a very high choline peak which appears in the prostate spectroscopy, which shows the appearance of cancer in the prostate. So this is again a very straightforward application of MRS in detection of cancer in prostate gland. And these are the two major cancers in men and women, the prostate cancer and the breast cancer. So very nicely one can acquire a spectrum from these lesions, breast lesions of the prostate region, and diagnose the cancer. Secondly, we also discussed that if you do a CSI, the chemical shift imaging, instead of selecting a single voxel, you select a huge area, and in the prostate, like it is shown here, you have so many voxels over here. Then you can see heterogeneity within the prostate, which are the places where you have cancer, where there are benign tissues. This can sometimes help the surgeon in dissecting out the cancerous. Mostly they remove the whole of prostate, but sometimes if they are very sure that the whole prostate is having spread these cancerous signals, then probably they can remove the complete prostate itself. Otherwise, they can look at the heterogeneity of the tumor in the prostate gland. Then MRS can be used in looking at the healthy aging, how brain deteriorates with aging, and this is a 1H spectrum for the from the posterior cingulate cortex, which is the location is shown here. This is a voxel. There are three-dimensional voxel, 1 by 2 centimeter cube, and then these are the spectra from the from the region for the healthy control, and also from the healthy young individual, and from the elderly person with more than 65 years of age. And if you look at the metabolite ratios, you can see glutathione. I told you last time that glutathione is a antioxidant in the brain. So with the aging, this glutathione is reduced in aging group compared to the healthy young individuals. And probably after a certain age, 45 or 50 years, one should start having antioxidants to avoid any further deterioration in the brain and in the cognitive functioning. Similarly, as glutamate, in the glutamate also you see a decrease in the glutamate signal. Glutamate is a excitatory neurotransmitter, and this is also reduced in elderly population compared to healthy young. Mannitol is another metabolite which is increased in this elderly population, because mannitol is a, as I told you last time, it is a sugar phosphate. It shows, it tells you about the deterioration in the brain, and you find mannitol increased in dementia as well as Alzheimer's disease very considerably. So there's an increase in mannitol also when the even if the brain is healthy aging is there, but still with aging, the process of aging, the mannitol signal increases. And similar is the total creatine, which also increases with change in age or increase in age. Similarly, one can select, one can select this voxel any part of the brain, and look at the metabolic profiles in that particular region. And we have done this study on posterior cingulate cortex as well as anterior cingulate cortex, which is located over here. And we have shown how these metabolic change. And then you can also show the correlation of these metabolic changes with the cognitive parameters. Maybe this study is still on. And then we also try to use some of the herbs like Brahmi Gripta, which is being given to elderly population to arrest this cognitive decline. And you can have you can repeat the MRS after maybe 3 months or 6 months of this herbal therapy, and see how these metabolites reverse or they are preserved at the current level. So this is a very good study which is very useful to the elderly population. Then this is a 1H MRS of the muscle, and this shows the intramyocellular and extramyocellular lipids in the muscles, and the concentration of EMCL and IMCL will tell you about the muscle myopathies and other disease conditions in the muscles. And suppose there is a muscle tumor or muscle cancer, you will see the total choline peak will get higher. This will become bigger, and these ratios will also change. So again, a very strong technique to look at the tumors of muscles, which sometimes becomes difficult in other situations, and you have a actually metabolic profile in the muscle, and if there's a tumor, you can place the voxel within the tumor and see what is the metabolic profile within that region. We all have heard about fatty liver, and this is another technique where you can look at the the fat contents over the liver compared to the tissues. So this here also you can calculate the, this 0.9 to 2.4 ppm are the most of the lipids fall in that region, and you can look at what type of lipids and how these lipids are creating the problems in liver, and how liver is getting damaged. And ultimately a person with fatty liver for a long time can convert into non-alcoholic liver disease, or this can also happen with the people who consume a lot of alcohol. They also develop cirrhosis, liver cirrhosis, which can ultimately be fatal most of the times. So this is another technique where you can show the fatty tissues on the liver, fat deposit in the liver, and after the person is made to if the person does some exercise and reduces his weight, then you can see how this fat water ratio will come down, because the fat is now being is being utilized to convert into energy. Generally, the neurotransmitters cannot be visualized by conventional techniques in MR spectroscopy. But if you use special techniques, special editing techniques in spectroscopy, like megapress technique, you can look at the GABA or glutamate, glutathione. All these neurotransmitters can have you can quantify them, and these neurotransmitters are at much lower concentrations, so they are not visualized on normal press and normal conventional spectroscopy sequences. So you need to have little more complicated techniques like megapress and spectral editing to calculate or compute the concentration of these neurotransmitters in brain, and they are very helpful in studying the brain functions and as well as brain disorders. So here we are showing this is a GABA mega press in a healthy and a these T2 diabetes patient. You can see how GABA is lowered in T2 this the diabetic patients compared to healthy control. You can see the this is a spread of these GABA values in the T2 in the diabetic patients, and in the healthy controls. Similarly, you can see here the glutamate levels are much lower in diabetic patients compared to healthy control. So you can actually see why these healthy, there's a slowing down, and excitatory systems are disabled or degraded in elderly population, or in the disease case due to the aging or the disease process. And so in short, the average level of GABA and glutamate was significantly lower in diabetic mellitus compared to healthy controls. And then you can also show, this is a correlation graphs of this is HbA1c versus glucose concentration. You can see there's a very high correlation between the two components, because we expect when there's a glucose levels are high in brain or in the blood, you expect HbA1c to be higher. So there's a linear correlation, positive correlation between the two parameters. And also you see a GABA GLX ratio also showing a positive correlation with HbA1c. That means when patient is severely diabetic, the GABA values also increase accordingly. So there's a direct increase of GABA levels when there's a diabetic mellitus. You can see here. And also, if you look at the glutamate, there's a inverse correlation between glutamate and the diabetic mellitus. So this you can also look at these these parameters, these metabolic parameters, and do the cognitive correlations also. One can do that, whether the memory is affected, or attention is affected, or the visual-spatial skills are affected. One can really do a correlation analysis on these metabolites and the cognitive functions. So here you can see this, as I told you, -II-L is always -II-L always increases whenever there's deterioration in the brain functioning. So you can here also you can see this is a diabetes mellitus type 1, not type 2. Here also you see the MI is increased compared to healthy controls, and you can see a positive correlation between MI and HbA1c. As the HbA1c increases, MI also increases. So it's showing a very strong 0.75 correlation coefficient, very strong correlation between MI and. So these people may also develop, if the diabetes remains uncontrolled, the dementia or the Alzheimer's may set in these patients. They are more prone to dementia and Alzheimer's disease. So I finished the proton one. Now we shift to the multinuclear spectroscopy, and I will discuss about elements other than proton, because proton we discussed in the last lecture. So so now when we say multinuclear spectroscopy, you are referring to the nuclei other than protons, and which may be helium, carbon-13, fluorine, sodium, phosphorus, and xenon. So all these nuclei can be studied using NMR, but some of the nuclei here are not do not have enough concentration and natural abundance in our body. So it is difficult to study some of them, but the mostly which are easily studied using in multinuclear spectroscopy is phosphorus 31 and sodium 23. These are the two nuclei which can be studied much easily, though the sodium 23 has a quadrupolar relaxation because it has a spin of 3/2, so it is little complex. It is a non-spherical charge distribution, and it has very short T1 and T2 times, making their signal a little difficult to detect. So here you see on the lower table you see the hydrogen. It gives you strongest signals, occurs in all biological molecules, and primary nucleus of interest for MRA and MRS. And best part is, if you are buying a imaging system, you do not need any special hardware for doing MR spectroscopy for hydrogen, because you have all the software and the hardware available on the imaging system to carry out such experiments. But if you want to do any elements other than hydrogen, then you need to buy special hardware which should cover the resonance frequencies of all these nuclei which are listed here. And if you look at the carbon-13, it gives you, I told you last time, the peaks are spread over a long chemical shift scale. So they are well resolved peak but weak signal and requires decoupling from hydrogen. And all these can be only done when you label substrate and give you inject the isotope to increase the concentration of the signals to signals so that you can detect these metabolites. Then fluorine 19 is frequency is very close to hydrogen. It is 40.06 megahertz per Tesla, which is very close to hydrogen. You can see hydrogen is 42.58 megahertz. It gives you a strong signal, but it does not naturally occur in biological tissues. So it can be used to label and measure drugs so it is essentially useful in drug metabolism. One can use fluorine 19. Sodium 23 has a spin of 3 by 2, and it has a much lower frequency, and sensitivity is also much less than hydrogen, but still it gives you a strong signal because it has natural abundance. But it is very useful to study the functioning of sodium pump, intracellular, extracellular sodium concentrations using sodium and sodium spectroscopy. And one could also generate a sodium image in the brain if you have appropriate hardware, the coil and the high Tesla magnet, so that you have enough signal to detect these signal from the sodium. Then 31p phosphorus, it has 17.24 megahertz gyromagnetic ratio, and it has 100% abundance in natural abundance, and of course sensitivity is again much lower than hydrogen, but you get enough signal from from phosphorus compounds, and it is important in monitoring energy metabolism. And here also peaks overlap over each other, but most of the peaks, most of the important metabolites you can still look at and quantify them. And xenon 129 has a spin of half, and it has 11.78 megahertz per Tesla this gyromagnetic ratio. If you do hypopolarized xenon, you can actually make people to inhale xenon, hypopolarized xenon, and this will enable MRI of the lungs, which is not possible otherwise, because the lung is essentially a air species. So you cannot do imaging of lungs by MRI otherwise. So, but if you make this xenon, hypopolarized xenon, this can be inhaled, and you can produce nice images from the lungs and look at these air sacs, whether they are they are intact or they are destroyed. So these are the nuclei which we discussed. I think and only thing I want to tell you that in fluorine 19 you can also do calcium ion concentration and other metal ion concentrations. You can do the temperature measurement and also partial oxygen pressure. And in sodium you can do transmembrane sodium gradient, as I told you, the intracellular extracellular sodium concentration, which can give you information about tissue and cartilage structures. And 31p is essentially cellular energetics. It tells you functioning of the mitochondria and how the energy products are being produced and utilized during the metabolic reactions. So if you look at the phosphorus is an important component of our body. It is about 1% of the weight of the body, and it is present in the form of sugar phosphate, and which is backbone of all the nucleic acids. It is part of polar group of phospholipids of cell membranes like glycerophosphocholene and glycerylophosphoethanocholine. I mean, these are the two major components which are the components of phospholipids, and you can obtain NMR signal only from small mobile phosphorus containing molecules, and the concentration of these molecules should be at, approximately 1 millimolar or higher. And so not, as I told you, not all metabolites fall in this range, but whatever metabolites are in higher concentration than 1 millimolar, one can look at them in the MRS. And when you talk about phosphorus at E1, the main signals come from ATPs, adenine triphosphate, phosphate, inorganic phosphate, phosphocreatine, and the weak signals come from PME and PD, which are the sugar phosphate and the phospholipid components. So they are at lower concentration, so they are seen, but the concentration being less, so peaks are much smaller, but still you can quantify them using P31 spectroscopy. So here you see a spectra from the calf muscle. You can see the PCR, which is a energy reservoir, and you can see the phosphodiester. This is inorganic acid phosphate, and over 7.5 here you have phosphomonoester. PME here will appear over here. Then these are the three, ATPs, gamma, alpha, and beta. I think most of you will be aware of this ATP structure. And if you look at there are three in ATP. There are three phosphorus atoms, and the structure is like this. And then each this phosphorus has only one adjoining phosphorus. So it will give a doublet. So you have doublet from gamma ATP and alpha ATP. You get doublets. But if you look at the central phosphorus, it has two adjoining phosphorus atoms, so it will have three peaks. So this beta ATP gives you a triplet, which is shown over here. You can see much more resolved triplet here and doublets here in these two alpha and gamma ATPs. So and there's a lot of other peaks which overlap with the gamma ATP and the alpha ATP. So if you measure the concentration of these ATPs, they may not reflect true ATP concentration. So in MRS, in vivo conditions, we always measure beta ATP, all the ratios with respect to. Whenever we will show you ATP ratios, they are with respect to beta ATP, because this beta ATP is free of any contamination from any other compound within the muscle. So all the concentrations which are being measured will be measured with respect to beta ATP. But we'll mention ATP there, but they are actually beta ATP, measured components. So this is a muscle, from other areas. This is a P31 spectrum from the brain, human brain. You can see different metabolites, which can be seen GPC and GP are able to resolve properly. Here you can see in phosphomonoesters, you can see phosphoethylcholine and phosphocholine. These peaks are also very well visualized because they are at higher field strength. You can see how beautifully these metabolites are visualized on brain. And this is a liver spectrum. And liver spectrum, we discussed last time, there's no phosphocreatine in liver, so you don't see much peak. This may be a contamination only from the skin part, so you see a very small part of phosphocreatine. But if you look at the brain, it has a very huge PCR peak. The liver doesn't have PCR peak, so you immediately know this spectra has come from liver. You can look at the different component, beta ATP, alpha ATP, NAD, UDPG, and all other peaks are very well visualized. You can see when you are doing a 7 Tesla magnet, if you look at the peak, you can see how beautifully all the peaks are being resolved. You see a perfect doublet from the gamma and alpha and a triplet from the beta ATP, very nicely visualized. Similarly, the phosphoethylcholine and phosphocholine are also very well resolved, and they can be now actually quantified when you work with 7 Tesla or higher magnetic field. Same thing is here. In the phosphodiesters, GP and GPC is well resolved, and one can calculate the area under these peaks and find out the concentration of these metabolites. Similar thing is with the inorganic phosphate, which is also very well visualized here. So this also shows if you have a stronger magnet, your signal to noise ratio is going to be better. Your visualization of metabolites is going to be better, and you can quantify your metabolites with more accuracy. So now, when we look at these these metabolites, what do they signify? And if you look at the adenosine triphosphate, as I told you, there are three phosphorus atoms, and corresponding to these three phosphorus things, you get three peaks. The chemical shift is The beta ATP is about minus 16 ppm, gamma ATP is minus 755 ppm, and alpha ATP is minus 2.5 ppm. And this is a principal ATP, you all know, it's a principal energy storage for this. And the ATP breaks down to give you PCR and ADP, and then when your muscle works, phosphocreatine, which is a buffer of energy, is utilized to for the function muscle to function. And J coupling for phosphorus to phosphorus is about 16 hertz. So you will have these peaks 16 hertz away from each other whenever you have these doublets or triplets. In brain, up to 25% of ATP signal comes from other nucleotide triphosphates, NTPs, including guanosine, uridine, and cytidine, triphosphates. So there are so many other, components which fall under these peaks. And ADP and AMP, adenomonophosphate resonances, they are too small to detect using in vivo MR spectroscopy. Now, phosphocreatine is taken as as I told you, say energy reservoir, and it is taken as a internal reference. All the peaks are measured with respect to PCR. So PCR will always appear at 0 ppm, and all the other peaks are measured with respect to PCR. And this is the largest peak in the spectrum in phosphorus, and it is absent in liver and red cells. As I showed you in the liver spectrum, PCR was absent. It is used as an internal reference for chemical shift. As I again told you, it is at 0. So all the metabolites are measured in respect to phosphocreatine. Then we have inorganic phosphate, which is called the acid phosphate. These are ionic reservoir for 31P in biological systems, and there's a exchange of uh, these two H2PO4 HPO4 components which keep exchange, but you actually measure average value. You measure actually a average peak in the spectrum, because these exchange rates are very fast. So when you measure the inorganic phosphate, you get a single peak, and this peak is pH dependent. And when there's a acidosis in the muscle, there will be a shift in the PI peak from the PCR, and you can actually calculate the estimate intercellular pH by using this technique. The next peak is phosphodiester. I've shown you all these peaks now, where they appear and what is their role is shown now here. A group of resonances in the delta of 2.5 to 3.5 ppm range, including a broad peak for cross-linked membrane phospholipids. So, and I told you it's grasophylline, phosphocholine, GPC, at 3.0 ppm, and it's a major source of choline storage in the cytoplasm and important renal osmolyte. Similarly, GPE is a smaller PDE peak just to the left of GPC at delta of 3.5. It's a membrane breakdown product. It is same as when we looked at the hydrogen spectroscopy, we looked at the choline peak, and whenever there was a tumor or any disease process, the choline concentrations were higher in tumors and other infections. Similarly, here also, when you do when you measure phosphorus spectroscopy in tumors and other infections and other things, you will find these GPC and GP raised in tumors and other disease conditions. You can because they are also depicting the same, but through phosphorus spectroscopy. Then phosphorus monoesters PMEs, and they are group of resonances in the range of 6 to 7 ppm, reflecting membrane synthesis, especially prominent in brain and liver spectra. At least two discrete peaks can be regularly identified, which are PE, phosphoryethylamine at 6.8, and phosphocholine at 6.2 ppm, and they are precursors of storage of the choline containing compounds. Then are the nicotine adenine dinucleotide, NAD and UDPG. These are again peaks which fall in between alpha ATP, almost as a shoulder to alpha ATP, and UDPG is shows a small peak in liver just right of NAD at minus 9.7 ppm. So these are some of the metabolites which are visualized in phosphorus adhesion spectra, and how what are their roles has been mentioned over here. And if you look at the most of the studies using P31 has been into either in muscle or in brain, and sometimes in monitoring the tumor therapy. It was used as a treatment response in tumor therapy. So most of the work is in muscle, and you can see here the and we I told you all the peaks are measured, and you can measure absolute concentrations, but for that you have to do a spectra for a very long time, because you have to wait for all the relaxation processes to be complete. So what we do, we measure here the ratios with respect to other metabolites, so that you're taking account of, suppose somebody has a lot of muscle mass, some have less muscle mass. So always, to neutralize for those effects, we use the ratios, and we see here PCRPI, which tells you about the oxidative stress or the oxidative metabolism. You can see this, and in most of the literature, in Western literature, they have shown the value of PCRPI to be 8.4 plus minus 1.4 as standard deviation. Similar other peaks, PCR-ATP 4.23 plus minus 0.24, and there has been large variation in both of PCR-ATP in both health and disease, and it can decrease up to 50% in some disease conditions where ATP production is is compromised. Then PIATP is again when there's a lot of acidosis happens in muscle because of exercise or because of the myopathies. The PI concentration is higher, so PIATP can decrease in various disease conditions. PI will increase, so PIATP will also increase compared to the healthy, which is 0.56 values AU. And then PD ATP is another very important parameter. PD tells you of the muscle fiber degradation. It tells you about that. So when there's a myopathies or any muscle degradation, dystrophy in the muscle, so you can find PD peak is increased. The concentration of PD increases. So this is a normal concentration of PD in healthy children, and this is slightly higher in adults. And this increases with age, can increase in some disease as much as, and sometimes it can increase 2 to 3 times in dystrophic muscle when there's a muscle dystrophy. So similar is PI-PCR. These are all parameters which we measure when we evaluate the muscle functions. And then there's a pH value. As I told you, you can measure the pH by measuring the shift of PI peak from PCR when the muscle is working hard, and when there's acidosis happening, and then you can measure the intracellular pH by measuring the shift. So you can actually measure intracellular pH by P31 spectroscopy, which is not possible otherwise, unless you inject a probe inside the muscle. But here is non-invasively you are able to calculate the pH of the muscle. Then you also look at the total PCR recovery rates, and this is very important. Particularly when these athletes, when they do when they do a lot of exercise, they run a long marathon or something, then it is important for them to recover. Their PCR is utilized, ATPs are utilized. So how quickly they can recover from that fatigue condition is very important. This is called PCR recovery rate. This tells you about the oxidative capacity of the muscle, which should be higher in athletes and sports person and healthy individuals. So you can measure the oxidative capacity of the muscle in health and disease conditions. So previous studies have shown that resting metabolic concentrations, you can measure P31 spectra during resting and also during exercise. You make the person to do some exercise on ergometer, and then you can measure the depletion of these metabolites because of exercise, and how muscle becomes acidic. You can measure using P31. So, but we in the beginning we all measure the resting metabolic concentrations. They're different between, and there are different fiber types in the muscle. Some people can run very fast for a very short distance, like sprint runners. They can complete the race very fast, but they can't run for a long time. But there are, these marathon runners, they can run for a longer time, they can't run faster. So there are different type of muscle fibers which are trained, and there could be a change in fiber composition by training. One can do that. So you always measure the PCRPI and PCR ATP ratios gives you the myofiber subtypes in humans. The lower PCR ATP and PCR-PI ratios and higher PI ATP seen in resting muscle of patients with genetic. If a person has genetic defect or mitochondrial oxidative phosphorylation defect, then you will have lower PCR ATP and PCR-PI, lower PCR-PI, but you will have higher PI ATP in the resting muscle. In muscular dystrophies, you will find a intracellular, intramuscular pH probably relates to membrane leakage and sodium accumulation. In some patients, there could be a multiple PI results suggest pH heterogeneity. So this is all what is from the previous literature. And increased PDE ATP ratios found always in muscular dystrophies and fibromyalgia and in elderly subjects. And there it is supposed to reflect elevated membrane turnover and disturb phospholipid metabolism. And we have also seen in many cases in Indian population, in healthy Indian population, we have found higher concentrations of phosphodiester peaks, which I will show you in the subsequent slides. So we have found that our muscle, Indian muscle, it looks like a very weak, bioenergetically looks like a much weaker muscle than the Western population, because most of the studies which are shown here are from Western population. The studies which we have done in India, I will show you a little later. So whenever there's an increase in PD, there's a muscular dystrophy or there's a disturbed phospholipid metabolism, and muscle is weaker bioenergetically. Muscle will not be able to work with very high efficiency. Then one can also measure free intramuscular magnesium concentration, which is you will find it is decrease in Duchenne muscular dystrophy. Like any dystrophy, this magnesium concentration is also decrease, which is a consequence of membrane leaking membrane or membrane leakiness. So all these studies one can carry out using. And now question is, how do we localize the MRI signal from the muscle? As I told you, muscle is a very superficial structure. You can use a simple loop of wire, which is at resonance frequency, and acquire signal from that. So what we call is the this surface coil. You can see this is a single loop of coil which can be placed directly over the muscle, and you can acquire the signal from the P31 signal from the muscle. Since muscles, you don't have muscle starts right from the uppermost layer, so you will always get a good signal from surface coils if you are doing the muscle spectroscopy. And most of the times these coils are very easy to build, and muscle doesn't need any specialized pulse sequence. Simple free induction decay pulse acquire, what we call, can be used to acquire signal from the muscle. And these coils are also much easier to build. They are simple LC circuits and tuned to the resonance frequency of phosphorus, and you can get signals very nicely from this. The only problem with surface coils is, as I told you earlier also, they must be placed close to the region of interest. And, then what happens, the signal reduces as you go away from the coil. But since you are measuring a spectrum, and, it doesn't matter, you are not measuring signal from a very selected area. You are measuring, signal from the whole of the muscle. So it doesn't matter even if you are not selecting a signal from a particular area. And there's no changing of ROA in this case. And it, as I told you, the signal profile goes reduces as you go away from the comb, so it has a inhomogeneous radio frequency field. When you transmit through it, it doesn't give 90-degree pulse equally at various depths. So you have to be, very careful in designing your experiments when you are doing, surface coil spectroscopy. So sometimes it is necessary. Suppose there's a tumor in the muscle, and you have to go to a depth. So at the most, one can use a dress sequence, which is sequence like this. You can see surface coil is here. The profile of surface coil is like this. It goes up to the diameter of the one radius of this coil. One can have this sensitivity of the surface coil. So what we do, we apply a radio frequency pulse, 90-degree pulse, only a slice stretching gradient. So we are selecting only a slice, and then from we take signal from this one of these slices. So it's very simple sequence. And but if you want to select a very small region, so now you will be acquiring signal from the whole slice. This slice, one particular slice which you have selected, and there are techniques available where you can select a voxel also. They are very time consuming and very difficult to implement on MRS. One such sequence is IS sequence, which was used for some time, but now it has been it has been given up because it was very complex and it used to take a lot of time in doing the measurement. So this is actually measured signal from a skeletal muscle from humans. You can see the PI, PDE, phosphocreatine at 0, and then gamma ATP, alpha ATP, and beta ATP peaks. So there are, the NMR spectroscopy of phosphorus is in muscle. It is basically, used to study the muscle bioenergetics during exercise. When you are making the muscle works, how does the muscle work? What is oxidative capacity of the muscle? How good is the muscle for a athletic event, or for a sportsman, or for as a soldier? One can look at the muscle profile, muscle bioenergetics using P31. Then you can also calculate the oxidative capacity on the basis of PCR dynamics. And as I said earlier, the healthy muscle, a good muscle, should be able to recover from the fatigue very fast. So this tells you about the oxidative capacity of the muscle. So oxidative capacity of the muscle should be higher for a healthy individuals, sports person, as well as good athletes. And one can also use P31 to look at the response to a particular therapy, and I will show you some examples how this has been utilized. So this is a arrangement you can make. Now, of course, now you get a very MR compatible ergometers where you can make muscle to exercise. But here we used a very local system. This was a assembly which we made, and you can put a foot over this, and this foot over this pedal. One can person can pedal this against a weight which is lifted here, and you can make this muscle, calf muscle, to work, to exercise, while the person is being measured for P31. And coil P31 surface coil is under this under this muscle which is placed here, and then you can make the person to pedal against this weight and for a long time. And then as the person keeps working, muscle keeps working, there will be a condition of fatigue, and then you can ask him to stop, and then you can look at the recovery process. So it was about resting. 4 minutes of resting was given to the person. Then he did 6 minutes of plantar flexion constant weight exercise, which is against this weight, and then we monitored them for next 10 minutes while he was relaxing or recovering. So this is the gastrocnemius muscle which we were interested in, which is also called the calf muscle. So you can see here the spectra at different time points. This is a resting spectra, and then this is when the person was exercising. And you can see how such a small PI peak, and when the person was exercising for a long time, you can see how PI acid phosphate is going up. And at the same time, you can see phosphocreatine more or less constant, because ATPs are constantly repeating phosphocreatine concentrations, and you can see even ATPs then are going down, and then you can see after the recovery process is over, after the exercise stopped, you can see the recovery again, PI is going down and comes to a normal after some time. So you can measure the PCR changes in concentration of PCR to make measure the kPCR or the oxidative capacity. Here it is showing how you can measure the pH of this muscle, and this is the formula for calculating the pH, and delta observed is the chemical shift of PI from the PCR. The difference between this peak and this peak, chemical shift of this is delta observed, and if you put this formula, and you can calculate the pH of this muscle in that particular situation. Then this is the recovery. You can see here. This is a resting state, values of phosphocreatine. And when you make percent exercise, the phosphocreatine goes down. And when there's a recovery process, slowly phosphocreatine comes up. Similar thing is pH. And then you can look at the ATP is fairly constant because it is being utilized. And constantly being built from the during the metabolism, oxidative metabolism. So you can see, and these are the stacked spectra shown over here. So this is the kPCR. You can measure the recovery. This is a resting, and then the exercise, and then recovery process. You can monitor these values continuously. So this is the resting values for PCR, PI, and PD, ATP in thyroid disorders. And this is a healthy controls. You can see PCR, PI is high, which is desired. But if you look at hypothyroid subjects, PCR, PI has gone down, as well as the PD, ATP is high. As these thyroid patients also have muscle weakness. They also have they get tired very fast. So you can see the phosphodiester is much increased in these patients compared to healthy controls. And then there's a condition called subclinical hypothyroidism, which is, these are the cases where the biochemical profile is same as controls, but the TSH values are slightly raised. It could be, from 5. If 5.5 is the normal range, then up from 5.5 to 10. So these are the subclinical hypothyroids, and these cases, the clinicians are always confused whether these should be treated at this stage or they should wait and watch to see whether they actually turn into hypothyroidism. So this is a clinical dilemma, and most of the doctors don't treat these subclinical hypothyroid patients till their PSH raises further, goes to 15, 20, 30, then only they treat them. Otherwise, they just wait and watch and see whether they are going come back to the normal situation or they are falling into the hypothyroidism. So these are the values spread data points of the patients and the healthy controls. So this is these oxidative capacity measurement in hypothyroid. Again, healthy control. These are the hypothyroids, and these are the subclinical hypothyroid. You can see the oxidative capacity of healthy muscle is much higher than hypothyroid, is much reduced, statistically significantly reduced. And you can see the subclinical hypothyroid also. They are reduced compared to healthy controls, but still better than the hypothyroid subjects. And then if you look at, this is the response to therapy. When these hypothyroid patients, they had lower PCR-PA ratio, when pre-therapy, you can see these are the values. And when they were given post-therapy 6 weeks, you see their PCR-PA ratio was recovering considerably. And then after 12 weeks of therapy, they all came almost in the healthy control range. So this shows how they are responding to the medication by P31 spectroscopy. And as I told you, these subclinical hypothyroidism, doctors are always, not very sure whether these people should be treated or not. The endocrinologist, of the institute, he spoke to us, can we do anything about this? Then we found very interesting that, in these cases, if the PD, we found that PD as a potential clinical marker for therapy in subclinical hypothyroid patients. We told them if the PD is reduced compared to healthy controls, then they can be treated, because all others were controlled parameters. Only PD is one parameter which we found subclinical hypertrophy was reduced compared to healthy controls. So we said whenever there's a reduction in PD, increase in PD, and then we should be able to treat them when the PD is increasing and when PCRPI is decreasing. But this is only parameter PD parameter which is increasing healthy compared to healthy controls. So we should treat them, and this is also similar to hypothyroid. You know, the trend is similar to hypothyroid. So they can be treated if the PD is high, is going high. So this can be a very good technique to assess the physical endurance in soldiers and sports person. You can find out who will be the good athletes by calculating their resting parameters and then looking at their oxidative capacity. One can determine the the efficiency of the muscle of a sports person or the soldier. And this is another thing, this uh, this is vitamin D uh, supplementation. Most of us, if you all of you go for a vitamin D test, all of you will find they are you are short of vitamin D in your blood serum. And we have found then about 70 to 80% population are falling below the levels of vitamin D. Though we are a very we a lot of sun in our country, but still most of us are vitamin D deficient. And in early 2000s, when I was working on healthy Indian population, I found that our muscle, as I told you earlier, we found that our muscle is intrinsically weak, that they have a lower PCRPI ratio and higher PD ATP compared to Western population. Then at that time, I could not, find out the reason. We thought it could be just because our protein intake is much less than the Western population. So we published this data, and simultaneously, one of my colleagues, who is an endocrinologist, he was working on vitamin D levels in Indian population. He told me that we are very deficient in vitamin D. Then we thought, was our muscle function reduced because of vitamin D deficiency? So we did a study where we found people who are vitamin D deficient, did their muscle profile, and then gave the supplementation. We saw very glaring results. Then after the supplementation, vitamin D supplementation, the muscle functions, they improve drastically after vitamin D supplementation. And you can see all these values, the vitamin D levels for average was 3.4, then they were raised to 26, 27. So you can see how when the vitamin D supplementation was given, how muscle function improved drastically. So maybe this is one reason why or the the athletic events we don't perform very well compared to Western population. So there's a move in between to have a national program on vitamin D fortification, like we had iodine fortification several decades ago. So this was a very interesting study, and this was again published in a good journal. This finding. So these are some of the spectrum. You can see spectrum from the skeletal muscle at 3 Tesla and 7 Tesla. You can see how beautiful when you go to 7 Tesla magnet, how beautiful spectra you get with much lesser noise, good splitting, and good visualization of the metabolites. And you can calculate these concentrations with much more accuracy as compared to 1.5 Tesla and 3 Tesla systems. This is a MR spectroscopy with the heart. You can look at the myocardium. When there's a heart attack, you can see these regions. There are 4 regions: the septal wall, the anterior wall, the inferior wall, and the lateral wall. All 4 walls. We have done the P31 spectrum. You can see wherever there's an infarct, wherever there was a heart attack, and the tissue was damaged, you see only very small metabolites. The spectra looks like a noise. You can see here lateral wall as well as inferior wall because there has been an infarction in these areas. But septal wall, you can see nicely phosphocreatine, all ATPs, PD, PME, and PI. You can see nice spectra. But since there is no viable tissue muscle here in these regions, so you see the metabolites are not visualized in these areas. So similar ways, sodium NMR, and as I told you, it is utilized to look at the intracellular, extracellular sodium. But unfortunately, both the sodium, intracellular, extracellular sodium, they resonate at the same frequency. So it is very difficult to visualize them by normal methods. So what you do, you use paramagnetic shift reagents, which tag to the extracellular sodium and make it shift from the actual position. These are the lanthanide series shift reagents for sodium, and by injecting this, what you find, the extracellular, without this shift reagent, there's a single peak from extracellular sodium and intracellular sodium. But when you give this shift reagent, you inject, you can see the extracellular sodium and intracellular sodium are distinct peaks. So now you can measure the concentration of the two peaks, intracellular sodium and extracellular sodium, and find out the functioning of the sodium pump. So I think this is again the infusion of the one of the shift reagents. You can see in different conditions how intracellular and extracellular sodium peaks are shown and concentrations can be measured. I think I finish with this, and I thank you all for patient hearing. In case there's any question, please you can raise.
Thank you, sir. So participants, any question?
You got my material? I sent it
When, sir?
today morning.
Good morning. I think I had to check. I didn't check afterwards.
Maybe I made it morning, but I forgot to send it. But I think 1:30 I have already sent it.
After that, I didn't check. I'll check it and I'll let you know.
And there are some questions also. You can read them, and then probably you can put them in 3-4 parts. You can send it to them.
Okay, fine. Okay, and any question, participants? I don't think any question. Doubt, yeah. Yes, sir, you want to tell something?
I hope you have participants.
Yeah, participants are there, but they're not asking anything.
Okay. Then it's okay.
So I think subject matter they are way too maybe.
No, I don't blame them. Teaching NMR and these things remotely is difficult.
Let them read the materials.
There has to be eye-to-eye contact.
That's true. And they have to read actually study materials.
Yeah.
Asking. Okay, sir. Let us hope they will go through the study materials. And
Okay.
Yeah.
Okay. Thank you.
Okay, sir. Then thank you very much. So we'll be taking.
Thank you. Yeah.
Wednesday.
Let's break. Wednesday.
Sir?
Wednesday and Friday. Yeah.
Wednesday. So Friday you would like to conclude, right?
Friday is the last lecture, 12th lecture.
Yeah.
Unless you want to.
According to schedule, Friday is the last day. So I think we conclude it. Okay,
Yeah.
fine, sir. And in between, if students are asking something, I'll be in touch with you.
Yeah. Thank you.
Okay.
Thank you so much.
Okay. Thank you.
Okay. Bye. Bye.
Bye.